This is part one of a two-part lecture on the introduction to genetics. Understanding genetics, you have to understand body's DNA. Remember that we've said all semester long that the DNA of every cell in your body, except the sperm if you're a male, the ova if you're a female, have the exact same DNA. The DNA is transmitted as cells divide by the process of mitosis, which produces two cells which are genetically identical to the parent cell. And ultimately, those two divide to form 4, and then 8, and then 16, and so forth, until ultimately you end up with the individual who has made up of a collection of trillions of cells, all of which have the exact same DNA, except where meiosis is occurring in the testes and in the ovary. DNA itself is a molecule made up of individual nucleotides being the basic structures that make up and combine together to form DNA, much like monosaccharides combine to form polysaccharides, much like amino acids combine to form proteins. DNA consists of nucleotide building blocks, and those nucleotide building blocks consist of a sugar, which is a ribose sugar, a deoxyribose sugar, a phosphate group, and then one of 4 nitrogenous containing bases. So there are 4 basic nucleotide building blocks. One with base A for adenine, one with base G for guanine, one with base C for cytosine, and one with base T for thymine. These bases are complementary because DNA is a double helix, which means it consists of 2 long chains of nucleotides, and the bases are complementary. The bases adenine is complementary with thymine. The base guanine is complementary with cytosine. The order of these bases in the DNA molecule allows for the coding for the proteins that will be made under the direction of that DNA molecule. A region of the DNA that codes for a specific protein through the development of a specific messenger RNA molecule is called a gene. Genetics is what we're going to talk about today and in the second half of this lecture, and it is the study of inheritance and characteristics that are passed along from generation to the next by the DNA of the cells. The genome is the complete set of all of the genes in an individual, and genomics is the field in which the body is studied in terms of how those genes interact with one another. So this is a diagram showing a basic outline of the DNA molecule. You have these 2 long chains of nucleotides strung together. This one has guanine, this one has adenine, this one has adenine, this one has cytosine. The complementary strand of DNA have complementary bases. So if this one is guanine, this one is cytosine. If this one is adenine, this one is thymine. If this one is thymine, this one is adenine. So DNA aligns itself up with 2 nucleotide strands with complementary bases forming the rungs, such as the rungs of a ladder. This allows for when the DNA makes a copy of itself or replicates, it basically peels apart, as you see here, and then new bases are placed on either side, thus producing 2 DNA molecules which are identical to the parent molecule, which is the first stage in cell division, which is called DNA replication. So the DNA of the cell consists of these long strands of DNA molecules. Those DNA molecules are complexed and organized in the nucleus of the cell with the help of what are called histone proteins. These are proteins that the cell has made that help to organize and align and keep the DNA where it should be so it doesn't get all knotted up. In each cell of your body, you have about 1.5 to 1.7 meters of DNA. And if you add up all the DNA in all of the cells of your body, you would have a strand of DNA long enough to make 50 round trips to the sun and back. Normally in the cell, the DNA is not pulled together into structures called chromosomes. Chromosomes only occur when the cell is getting ready to divide and the DNA has made a copy of itself. Normally, the DNA is spread evenly throughout the nucleoplasm, the area inside the nuclear envelope, as what is called chromatin. Chromatin is that uncomplexed or unpulled together DNA, so that the DNA can be utilized to unzip in certain areas, making messenger RNAs, allowing proteins to be produced that cell has directed the DNA to make. As we study the introduction to genetics, what we're going to find is that not only does it give you an understanding of how inheritance occurs, but we also will learn that there is the power of prediction. You have the ability to predict the outcomes of certain crosses between individuals based on the genetics that those two individuals have. Knowing how those genes are distributed during meiosis allows for certain combinations to occur in a percentage of the offspring, and we'll talk about calculating the probability of certain traits in the end of this lecture. The way in which genes are transmitted from one generation to the next can be several different types, but in general they are together called modes of inheritance. As we said before, every cell except the sperm and the ova has the same DNA in its nucleus. And that DNA, when it complexes together at the time of cell division, complexes together to form chromosomes. Chromosomes are the complex DNA of a cell that's getting ready to divide. That's the only time chromosomes appear. Normally, the DNA is spread evenly throughout the cell as chromatin. But when it pulls together as chromosomes, you can find that what there are a total of 46 chromosomes in the human genome. But rather than thinking of it as 46 chromosomes, I want you to think of it as 23 pairs of homologous chromosomes. What does that mean? That means 23 pairs of chromosomes that code for the same traits. And you got one of those from your mother and one of those from your father. When you take a picture of those chromosomes and line them up by homologous chromosomes, you end up with a structure that looks like this. That's called a karyotype. A karyotype is an actual pictured representation of the chromosomes. So instead of thinking that the DNA of your cells is divided up into 46 chromosomes, think of it as it's divided up into 23 pairs of homologous chromosomes that code for the same traits. But even being more specific, you actually have 22 pairs of what are called autosomes, which code for traits, and one pair of sex chromosomes. And those sex chromosomes are going to determine your sex. In this case, this is a female because there are two long X chromosomes. If it was a male, you'd have a long X chromosome and a very small Y chromosome. So this is homologous chromosome pair 1. Again, you got one of those from your mother, one of those from your father. Chromosome pair 2, you got one of those from your mother, one of those from your father, and so forth and so forth. Each of these chromosomes have in their DNA strands regions of the DNA that code for specific traits. And we'll talk about some of those today. But first we got to talk about Gregor Mendel. Gregor Mendel lived from 1822 to 1884. He is considered the founder of genetics. Gregor Mendel's story is a sad story. This was a man who grew up on a farm, and he was curious and was sent off to college, and all he ever wanted to do was teach biology in high school. And at the time, he had to take a series of tests to get to that, and he got to the very last test and he failed. And at that point, he could not teach biology. So he ran out of money. He didn't know what to do, so he joined the church. And the church sent him back to college. And again, he went to some more classes. And again, he took the test to teach biology in high school. And again, he failed. So Gregor Mendel at this point now wanted to teach biology, was not able to do that. But he was now a member of the church. So he was placed in a monastery, and he was given jobs to do, but he also continued to want to work on inheritance. For years, farmers had understood that you can breed certain breeds of cattle and whatnot to produce better meat, but they didn't know how that happened. And he wanted to study those things. So the first thing he did, since his upbringing in the farm where he grew up, they did beekeeping, was he wanted to do experiments with bees. And that did not work out because the bees that he was utilizing kept stinging the other priests, and they told him he couldn't do that. So then he wanted to do mice, and the church said, no, we can't have you having these mice, basically having sex and doing reproductive experiments on mice. The church would not tolerate that. So Gregor Mendel said, well, what about if I do plants? And the church was okay with plants, so he picked, in general, different types of plants, but he's most famous for studying various characteristics of pea plants that he would grow. And pea plants had certain characteristics, as shown on this chart: the colors of the flowers, the positions of the flowers, the height of the plant. Some were taller, some were shorter. And he would cross-pollinate these plants and then look at the offspring produced by them. And because of that, he was able to derive the fact that he could predict the statistical outcome of cross-pollination of these pea plants by knowing what characteristics the parents had. And he studied and he published his data. Unfortunately, since he was not a college professor or anybody highly admired in the worlds of science, they did not believe him. And he died in 1884. About 40 years after his death, somebody actually reviewed his paper, and at that point, they did further studies, and they proved that what he was studying was correct. And to this day, he is revered as the founder of genetics, or what we call Mendelian inheritance. Making his story even more tragic, after this occurred, later on down the road, when he was proven correct, other scientists tried to repeat his experiments and were not ever able to get data as good as his, insinuating in many circles that he actually fudged some of his data, leading to the term the Mendelian paradox, or the too good to be true paradox. So this poor man, all he wanted to do is teach high school science. He founded a science that we still use to this day, and he's still being maligned for it even after his death, because they're saying that his data was too good to be true. So I want you to know Gregor Mendel as the father of genetics, because he worked hard for that title and never really got the credit for it, either before or after he died. Several terms we're going to talk about today. First of all, we've already mentioned a gene. A gene is a region of the DNA that codes for a specific trait, or more specifically, codes for a protein to be produced that can lead to a specific trait. There are variations of genes. Not all of us have the exact same genes. An allele is a variation of a gene. We'll talk about different alleles. understanding that for each homologous chromosome, you're going to have a gene for the same trait or protein. One came from your mother, one came from your father. A homozygous individual is an individual that got the same allele from their mother as they got from their father. A heterozygous individual is an individual who got one allele for a gene from their mother and a different allele for a gene from their father. This will make more sense in a minute here. For example, we look at a single allele trait such as earlobe shape. And here we see two earlobe shapes. Here we see a free earlobe, and here we see a fixed earlobe. And you either have a free or a fixed earlobe. And there is one allele that controls whether it's free or fixed. And we'll talk about how that all happens. But you got one allele from your mother on a homologous chromosome and one from your father. Other terms that you need to know. Genotype. Genotype are the actual alleles for a gene. So if you got the same allele from your mother and from your father for a gene, you'll have the same allele for your genotype. You'll have two of the same alleles. A phenotype is the observable trait. That is whether or not you have a fixed or a free earlobe, or whether or not you have a widow's peak or not, or whether or not you can roll your tongue or not. The observable trait is the phenotype. The genotype are the specific two alleles for that trait. Different types of inheritance patterns exist, but the most common one is a dominant recessive inheritance pattern in which one of the alleles masks the other one if you have a heterozygote. What does that mean? That means if you got one allele from your mother and another allele from your father, the allele that is dominant will display that trait. In other words, you will look the same if you got one allele from your mother and one allele from your father as the dominant allele, as you would get if you got two dominant alleles. They look exactly the same phenotypically. A recessive allele is masked by a dominant allele, which means the only way you can phenotypically describe or display that trait is if you got one of those from your mother and one of those from your father. We go back to earlobes. The allele for free earlobe, or big E, is dominant. The allele for an attached earlobe, or little e, is recessive. Which means that if you have a free earlobe, your phenotype is you have a free earlobe. But your genotype could be big E, big E. You could have gotten two big E alleles from your mother and your father, or you could have gotten one big E from one parent, one little e from the other, because the recessive will be masked by the dominant, and these will look exactly like these. So if you have a free earlobe, you know what your phenotype is. You have a free earlobe, but you do not know your genotype because you could be either one of these. They look the same. If you have an attached earlobe, since that is a recessive trait, the only way you could have gotten that is if you got one recessive allele from your mother, one recessive allele from your father, and therefore you have both recessive alleles, which allows for the production of the fixed earlobe. Does that make sense? Don't you miss hearing me say that? So, genes are regions of the DNA that code for specific proteins, ultimately leading to, in many cases, specific traits. If you have the same alleles, you are homozygous for that allele, for that gene. If you have two different alleles, you are said to be heterozygous for that gene. The genotype is the individual alleles, one from your mother, one from your father for that gene. The phenotype is the actual physical appearance, makes up the phenotype. What you see. So a dominant recessive inheritance pattern in the heterozygous condition, like we just showed, one allele dominates the phenotype. In other words, if you are big E, if you got two dominant alleles from one parent, one from the other parent, or you got one dominant allele and one recessive allele from the other parent, you will still have a free earlobe. In other words, these two look the same because this one is completely masked by the dominant allele in a dominant recessive relationship. The recessive allele will only be expressed if you got one from your mother and one from your father. We are going to talk also about conditions or abnormalities or situations in which there are abnormalities of the autosomal DNA. In other words, the 23 pairs of autosomes. In the second half of this talk, we will talk about sex-linked conditions, conditions associated with the sex chromosomes. What are examples of single gene traits? Well, we've already mentioned unattached and attached or free and fixed earlobes. And we said that free earlobes are dominant, so the genotype could be either one of these. Fixed earlobe is recessive, so you have to have this genotype to have a fixed earlobe. Freckles is another single gene trait. The presence of freckles is dominant, which means if you got one or both of the freckles dominant alleles, or big F, from one of your parents, you will have freckles. But if you got both of the recessive genes, one from your mother, one from your father, you will have no freckles. Being able to roll your tongue is a dominant trait. Now all of you are probably trying to roll your tongue as I say this. So if you can roll your tongue, which most of you probably can, you either have a genotype of big R because it's the gene is dominant, or you got one dominant and one recessive trait from your parents, which you can still allows you to roll your tongue because this is the dominant allele. If you cannot roll your tongue, you know what your genotype or your phenotype is. You can't roll your tongue, and your genotype must be little r, because if you had a dominant allele, you would be able to roll your tongue. Dimples is another one. The presence of dimples is a dominant allele. The absence of dimples is a recessive allele. So if you have dimples, then you either have two dominant alleles, one from your mother, one from your father, or a dominant and recessive allele, one from your mother, one from your father. But if you have no dimples, you must have gotten the recessive alleles from your mother and your father, because that's the only way you can have no dimples. And finally, the widow's peak hairline. The widow's peak hairline, or that pointed peak of the hairline in the front, is the dominant allele, which means that if you have a widow's peak hairline, your phenotype is widow's peak hairline. The genotype is either W, big W, big W. In other words, you got two dominant alleles from each of your parents, or a dominant and recessive allele. And since the recessive allele is recessive, the dominant will mask it, and you will have the widow's peak hairline. If you have a straight hairline, then you must have gotten both recessive alleles from your parents, because if you'd gotten a dominant allele, you would have a widow's peak hairline. Understanding how this works, and the work that Gregor Johann Mendel did, allows us to predict the probability of offspring if we know the genotypes of the parents. And you do this by producing what's called a Punnett square. A Punnett square is basically a square with 4 quadrants in it, and each quadrant is going to reflect 25% of the children that is produced. And then on one axis you put the mother, and on one axis you put the father and their genotypes, and then you fill it in, and each quadrant again is 25% of the offspring. So we're going to talk and give some examples. We're going to talk about widow's peak. We'll use this as the example. The presence of a widow's peak is a dominant allele, which means someone with a widow's peak, which is shown here, either has to have two dominant alleles or a dominant and a recessive allele. These people will all look the same. They'll have a widow's peak. If you have a straight hairline and no widow's peak, that is a recessive trait, which means the only way you can have that is to have a genotype of little w, little w. In other words, you got a recessive gene from both your mother and your father to allow for that straight hairline. These next slides are slides that are not on your initial handout. I want you to look at these. You may want to make notes about them because you will be asked on the exam to produce Punnett squares and predict the outcomes of crossing. It's not that hard to do. Basically, a Punnett square consists of a box with 4 squares in it. Each square is going to represent 25% of the offspring. You put one parent on one axis. In this case, we're putting the mother on this axis, and we're putting the father here. So in this example here, the mother has a widow's peak, and she's homozygous for that. What does that mean? That means she has big W, big W. So you put her 2 alleles on up here. And we're saying the father has a straight hairline. The only way you can have a straight hairline is to have 2 recessive alleles, or little w's. Once you do that, then all you got to do is fill in the boxes. And what you'll end up with is each box represents 25% of their children. I did this one first because if you fill in all the boxes, what you find is all of the boxes end up with the same genotype. They all end up big W, little w, which means 25, 50, 75, 100% of their children are all going to have the same genotype. They're all going to be big W, little w, which means they are all going to look like their mother. They're all going to have a widow's peak. None of them will look like their father, but genotypically, they will be both different than their mother and their father because they will all be heterozygotes. Does that make sense? Different example. Now the mother has a widow's peak, just like she did there, but this time she's heterozygous for that widow's peak. What does that mean? That means her 2 alleles are big W, little w. Let's say one of these, okay, is the mother. So this is a woman with a widow's peak who, again, has children with a man who has a straight hairline. He has little w, little w. So now you got big W, little w, little w, little w. You fill in the boxes and you end up with big W, little w here, big W, little w here, and then 2 little w, little w's here. So if each one of these represents 25% of their children, and these 2 are the same, and these 2 are the same, then 50% of their children will phenotypically look like their father and have a straight hairline because they'll have 2 recessive alleles. 50% of their children will look like their mother and have the same genotype as their mother. So the children that they have that have a widow's peak are going to have the same phenotype as the mother and the same genotype as the mother and represent 50% of their children. The other 50% of their children will both have the phenotype and the genotype of their father, have a straight hairline. Two more examples. Now, the mother is homozygous for a widow. Mother's got a widow's peak again. She's homozygous for it, like we saw on this example. But the father also has a widow's peak, and he's also homozygous. So that means the mother's alleles are big W, big W. The father's alleles are big W, big W. You fill in the boxes, and all 4 boxes have big W, big W. In other words, if the mother is homozygous dominant, the father is homozygous dominant, every one of their children will be homozygous dominant. 100% of them will be phenotypically similar to the parents and have a widow's peak, and genotypically be the same as the parents with 2 dominant alleles. You could also make the same rationale if the mother and the father both had a straight hairline, because then it'd be little w, little w, little w, little w, and you'd have all little w's in here. So if a mother with a straight hairline has children with a father with a straight hairline, all of their children will have a straight hairline because there's nowhere for them to get a dominant allele. Final example over here. Mother has a widow's peak. She's heterozygous, which means she has a dominant and a recessive allele in her genotype. And the father also has a widow's peak, but is also heterozygous. So he is also genetically big W, little w. So you have father and mother, both have widow's peaks, but they both carry a recessive allele. You fill out the boxes here, and what you end up with is this box and this box. 2 of the 4 boxes, or 50% of their children, are going to genetically be the same as their parents. They're going to have the same genotype. They're going to be heterozygotes. 25% of their children are going to have a genotype of big W, big W. And 25% are going to have a genotype little w, little w. Phenotypically, what those children are going to look like, these 3 boxes are going to look the same. They're all going to have a widow's peak. So 75% of their children are going to have a widow's peak. 25% of their children, or 1 box, are not. So we can utilize Punnett squares to predict the outcomes of crosses between individuals and predict the outcome of traits in their children. There are also some diseases that are autosomal recessive. What does that mean? That means that to get the disease, since it's recessive, you have to get the allele from both parents. You have to have both of the alleles to get the disease. Tay-Sachs disease is a autosomal recessive disease on chromosome 15 that causes abnormal proteins buildup in the brain, and it is almost universally fatal by age 5. Children born with Tay-Sachs disease rarely live beyond childhood. Cystic fibrosis is also autosomal recessive disease on chromosome 7. And it is more survivable, but does cause serious problems. It is a chromosome that produces a protein that inserts itself in the membranes of cells and allows chloride ions to escape the cells. What does it do? If you don't allow chloride ions from the inside of the cells to outside the cells in certain secretory cells, the mucus gets very, very thick and causes all kinds of problems related to that. This is an example of cystic fibrosis. Basically, it is a disease of chromosome 7, where if it is present in both alleles, you get an abnormal messenger RNA produced, which is going to produce an abnormal membrane protein, And that abnormal membrane protein is not going to allow chlorides to go from inside the cell to outside the cell, which would thin the mucus in certain areas. What kind of areas of the body are important for people with cystic fibrosis? It certainly affects the mucus in the airways, causing clogging of the bronchi, respiratory infections. It can block liver ducts. It can definitely block the pancreatic duct, causing problems related to the pancreas and inflammation of the pancreas. It can cause blocking of the intestines. It can cause abnormal appearance of the or abnormal absence of the ductus deferens, which leads to sterility in the male. And it causes a increased salty sweat, increased sodium in the sweat, which is one of the ways in which cystic fibrosis is diagnosed through what's called a sweat test. Microscopically, what's happening in persons with cystic fibrosis? Well, if this is a normal secretory cell, this is the inside of the cell, the cytoplasm, this is outside the cell. Normally, the normal gene produces a protein that allows chloride ions to pass from inside the cell outside, which draws water out, which thins the mucus outside in the airways, in the pancreatic ducts, and whatnot. But in cystic fibrosis, and there's several different actual abnormalities, but in both of them, what happens is the protein that is produced is abnormal, so the chloride ions can't get out, so the mucus that's outside becomes very thick, very sticky, plugging up the airways, plugging up ducts of the pancreas gland and things like that, and causing all of the serious problems related to the disease cystic fibrosis. Since it's a recessive gene, you have to have both of the alleles to get the disease. So in this example here, here you have two parents, mom and dad. Mom and dad, neither of them have cystic fibrosis. They are both absolutely normal, except for the fact that they are both carriers, which means they are heterozygous for that trait. Heterozygous state, the plus is genetically called wild type. That's the normal allele. So dad's two alleles are plus and the abnormal allele or cystic fibrosis allele. And mom's is the same. So since they're heterozygous and since the normal gene is dominant, both mom and dad will be absolutely normal and will not have cystic fibrosis. But if they have children and you look at and you make up a Punnett square, here's mom, here's dad. Basically, this is the normal allele. This is the cystic fibrosis allele. And what you will get when you do this cross is you will see that 75%, or 3 of the 4 boxes, of their children will have no disease and will be absolutely free of cystic fibrosis. But of those, 2/3 or 50% of all their children will be like the parents and will be able to pass the disease on as a carrier to the next generation. This 25% is completely free of disease and will not be able to pass the disease on to another generation. So 25% of their children will be genotypically normal, have no disease. 50% of their children will be also normal, but will be carriers like the parents. And 25%, or 1/4 of their children, will get both recessive alleles and will be cystic fibrosis patients. So when you look at making a pedigree, and we'll talk about this next lecture, these basically, the squares represent males, the circles represent females. They've given them names, but basically this would be Joe, and because half of it is dark and half of it is light, that means he is a heterozygote for this condition. This is Mary. Half of it is white, half of it is dark, the dark being the disease, the white being normal. And they have children. In this case, they had 3 children. And one of their children, Bill, is absolutely free of disease. That means Bill got lucky and got these 2 alleles. Their next child, Sue, is a girl who is one of these, so she is also free of cystic fibrosis, but she has the allele as a carrier, and she can pass it on to her children. And their third child, Tina got both of the cystic fibrosis alleles, and she has the disease cystic fibrosis. So this is an example of a autosomal recessive disease that's transmitted in many cases from carriers that don't know they carry the disease, like these parents were, and then all of a sudden a child shows up with the disease. Less common, but in many cases more serious, are autosomal dominant diseases. These are diseases in which if you get one of the alleles, you get the disease, as opposed to an autosomal recessive disease where you have to get both of the alleles. Several of these diseases are very serious, one of them being Huntington's disease, which is a disease on chromosome pair number 4, which causes an abnormal buildup of proteins in the neurons, especially in the brain. It causes severe debilitation and death. And the problem is that with many of these patients, they do not show symptoms until they're in their 30s or their 40s. It takes long for this to build up, and by then they may have actually passed the disease on to children that they have had. Marfan syndrome is another autosomal dominant disease. It's on a different chromosome, pair 15, and it causes a defective protein in the elastic fibers. We'll talk a little more about Marfan syndrome, I believe, in the next hour. So this is a example of Huntington's disease, and again, we show in this example we have this patient here. He has the disease, but he may be younger. He's not showing any symptoms of it yet. He is heterozygous for it. He has one normal allele and then the Huntington's allele, but the Huntington's allele is the dominant allele. So he will get the disease, but he doesn't know he has it. And before he finds out that he has the disease, he has children with this individual who has no disease. So she has two normal alleles. So this is mom. She has two normal alleles. This is dad. He has one normal and one Huntington's disease allele. And you fill in the boxes. And what you end up with is a situation where 2/4 or 1/2 of their children will be absolutely normal. The other half of their children will have Huntington's disease, because both of these, Huntington's disease being a dominant allele, if you get that dominant allele, you will have disease. So these 2 of the 4 boxes represent 50% of their children will have the disease, 50% of their children will be free of the disease. So if this is Dan and this is Ann here, and Dan has the disease, and they should show him as a heterozygote because he's heterozygous for it. What you end up with is they have 2 children, and Pam, their first child, is free of disease, but Eric, their second child, actually has the disease and will develop symptoms later in life. So far we've talked about dominant recessive relationships. We've said that some alleles are dominant, some alleles are recessive, and that's appropriate for most genetic relationships, but some are not. There are 2 other types of relationships that we need to talk about. One is incomplete dominance. We've said before that in a dominant recessive relationship, the heterozygote looks like the dominant allele. For example, in the free or fixed earlobe, if you were heterozygous for that condition, you have a free earlobe because it's the dominant condition. In an incomplete dominance, the heterozygote is an intermediary between the other 2. In other words, the heterozygote looks different than either of the alleles. It looks like a blend of the 2. These aren't nearly as common. Familial hypercholesterolemia is an example of an incomplete dominance where there are two alleles. There's the normal allele and the abnormal allele. And if you get two normal alleles, then your cholesterol level is largely normal. If you get two of the abnormal alleles, your cholesterol level tends to be super high and very dangerous. But if you get one normal and one abnormal allele, you're heterozygous, your cholesterol level is higher than normal, but not as high as these people. In other words, incomplete dominance creates a situation in the heterozygote that is between the two conditions. Probably the easiest example that I have of incomplete dominance is hair type. Hair type, there are two alleles for hair. There is the straight allele and there is the curly allele. And if you get two straight alleles, your hair is straight. And if you get two curly alleles, then your hair is curly. But if you get one straight allele and one curly allele from your parents, you end up with an intermediary called wavy hair. So wavy-haired individuals are heterozygous. They have a straight allele and a curly allele. This is another example. Again, you do not have this on your slides. You might want to write this down because this is an example of a Punnett square having to do with incomplete dominance. And here's an example of a woman, a mother has wavy hair, so she has to be a heterozygote. She has to have a straight allele, which is big S, and a curly allele, which is big C. She has to have those two. There's not a big one and a little one because this is an incomplete dominance situation. And the father also has wavy hair. So his alleles are one straight allele from one parent, one curly allele from his other parent on his homologous chromosomes. In this situation, if you fill it out, what you'll end up with is phenotypically, you'll end up with 25% of their children, remember both parents have wavy hair, 25% of their children will have straight hair. 25% of their children will have curly hair. And the other 50% of their children will have wavy hair like their parents had. So genotypically, you're going to get 50% having a heterozygous condition between the two alleles. 25% you're going to get homozygous straight hair, and they'll have straight hair, and 25% of the alleles are going to get homozygous for curly hair and have big C, big C. So if I was to give you a problem having to do with incomplete dominance and individuals with different hair types, if I was to give you a father with straight hair, you would have big S, big S. If I would give you a father with curly hair, you would have big C, big C. So it just depends on what criteria I give you, and then you fill in the boxes and then look and see what you got. Sickle cell anemia is another example of a disease that has incomplete dominance. It has a normal allele, which we're going to say is the big S, and it has the disease allele, which we're going to say is little s. And again, in a normal individual who does not have sickle cell anemia, you will have two big S alleles. That's what you have. If you have a person with sickle cell disease, you will have two of the disease alleles. But if you have a heterozygous state where you have one of these and one of those from each of your parents, you have what's called a sickle cell trait, which is not as bad as a disease, but it's not normal. If you're under certain types of stress, your red blood cells can still form those banana-shaped sickles, but you're much, much less likely to have severe problems than someone who has the disease. Again, here's an example of a Punnett square related to this. Here is a mother and a father, both of which have sickle cell trait. They have one sickle cell allele and one normal allele. So here's father. We put the father up on the top this time. He's big S, little s. And the mother, little big S, little s. You fill out the boxes, and what you get is 25% of their children, one box, are normal. 25% of their children have the disease, and 50% of their children have the trait like mom and dad had. That makes sense. The final genetic inheritance pattern we're going to talk about is codominance. Codominance means that in the heterozygous state, both of the conditions are expressed. Now, obviously, you cannot have both a fixed and a free earlobe. You cannot have both the ability to roll and not roll your tongue. But there are certain genetic alleles that can be present. The classic example of that is the alleles allowing for the A and B antigens to be produced on red blood cells. If you have type A blood, then your genotype has to be either big A, or big A, O, because there is 3 alleles in the ABO blood typing. There's A, and there's B, and there's O. O produces no proteins on the red blood cell membrane. So people with A, type A blood can be AA or AO. People with type B can be BB or BO. People with type AB have to have the genotype AB, which means they have to have gotten one of those alleles from their mother and one of those alleles from their father. And people who are type O must have the genotype OO, because if they had either an A or a B, then they would have those antigens on their surface of their blood cells. Codominance means that in the heterozygous state, such as this one, you produce both alleles. You get both A and B antigens on the red blood cell. You can see over here, if you have type A blood, you have A antigens. If you have type B blood, you have B antigens. If you have type AB blood, that means you got both of the alleles for A and B, which means you have both antigens on your red blood cell. And if you got 2 O alleles, then you have neither. And you can do Punnett square equations just like we did before with ABO blood typing. If I give you mothers and fathers genotypes, you should be able to fill out a Punnett square and figure out what the percentage of the blood types in their offspring would be. There are certain factors that can affect the expression of genes. In other words, not just having the genes doesn't necessarily mean they're going to be expressed. Certain terms, penetrance, expressivity, and pleiotropy, are basically distinctions that apply to if you have certain alleles, are they going to be expressed? And we'll talk a little bit more this hour and the next hour about that. Complete penetrance means everyone with the trait gets the condition. For example, if you get 2 dominant alleles for a free earlobe, you're going to have a free earlobe, because earlobes is complete penetrance. Everyone who gets those alleles gets the disease. And that's true for most of the examples we've used today. But there are some conditions that have what's called incomplete penetrance, which means that even if you get the alleles for a condition, it doesn't mean you always get the condition. The example for that is one of the genetic markers for breast cancer is called the BRCA1 gene mutation. And women can be tested for that to see if you have that mutation. If you have that mutation, you are 80% chance that you will develop breast cancer during your life. In other words, it increases your risk of breast cancer, but it's not 100%. In other words, there's not complete penetrance. If you have it, it doesn't mean you will get breast cancer. It means you are at tremendously increased risk. These women have to be watched very, very closely for signs and symptoms of cancer. And then there's variable expression, which means that if you get the alleles, you get different phenotypes. For example, there's a condition called polydactyly. Poly means many, dactyly means digits. And basically, people with polydactyly can sometimes have 2 extra digits, can sometimes have 3 extra digits, because the expression of having that, the phenotypic expression is variable if you get those alleles. Pleiotropy. Pleiotropy is a condition where you get a genetic abnormality that doesn't just affect one thing. For example, the ability or lack of ability to roll your tongue. That is just a single symptom that you get, a single phenotype. You can either roll your tongue or you can't. But there's some genetic conditions that can affect many different parts of the body. Marfan syndrome, as we've mentioned before, is an autosomal dominant defect. It affects the elastic tissues in your body, and it affects many, many different parts of your body. It can cause abnormalities of your heart, abnormalities of your major vessels. It can cause abnormalities of your skeletal system. It can cause abnormalities in your body shape. In other words, having this condition affects many different parts of your body, and that is what we discuss. That's what we call pleiotropy. Genetic heterogeneity. What does that mean? That means the same phenotype results from the actions of different genes. For example, there is only one pair of alleles that controls whether you can roll your tongue or you can't, or only one pair of alleles that controls whether or not you have a fixed or a free earlobe, or whether or not you have a widow's peak or not. But there are other conditions that are dependent on many different genes together. For example, hereditary deafness. There are several different alleles that can become abnormal that can cause deafness. For this example here, they give you 2 pairs of alleles. They give you the B allele and the R allele, okay? And it's a dominant recessive relationship. So if you have the large B allele, that's normal. The small b is deaf. So if you get 2 small b's, you're deaf. If you get 2 small r's, you're deaf. But if you get, so for example, this individual here has 2 small r's, so they will be deaf. This individual has 2 small b's, so they will be deaf. So if this is a deaf male, because this is square, and this is a deaf female regarding these traits, and they have children, the possibility exists that they are going, this one is going to give a dominant allele, and this one is going to give a dominant allele here. So their children, 100% of them will not be deaf, even though both of the parents are deaf, because of the fact that they have 2 alleles, and this one is dominant for the normal allele. Now, if this one was heterozygous for this allele, then that would be a different story. But in this specific situation where the father is deaf because he has 2 little r's here, the mother is deaf because she has 2 little b's here, well, she's only going to give a big R, and he's only going to give a big B, because that's all he's got to give in that allele. So all of their children will be heterozygous for both. They will all be not deaf, but they will all be carriers that they can pass it on to the next generation for both of those alleles. And finally, multifactorial traits. What does that mean? That means that many conditions are based, like we just mentioned, not on one allele, but on several alleles. And in many cases, it's not only the multiple alleles, but it also can have to do with environmental factors. Polygenic traits allow for a more wide distribution of phenotypes. For example, you can only have a fixed or a free earlobe. You can only roll your tongue or not roll your tongue. You can only have 4 different blood types. But in polygenic traits, where there's multiple genes acting on, you can to produce a response, you get a wide variety of phenotypes available. This is in conditions that you see that have much variability. For example, height. We are not all 1 or 2 heights. We are many different heights. Skin colors, eye colors, those are all things that have many different variations, and it's because they're polygenic in nature. There's multiple genes. We'll have some examples of that in a minute. In addition, multifactorial traits means that some traits are are influenced by environmental factors. For example, people who live in conditions where they don't get enough food, even though they may be genetically to become a certain height, they don't ever attain that height because they don't have enough fuel to make them grow taller. Certain diseases like heart disease, type 2 diabetes, hypertension, and certain cancers are multifactorial. For example, if you eat a lot of cheeseburgers and fats, you're going to increase your risk of heart disease, irregardless to what your genetic makeup says. This is an example of how heights are a polygenic trait. You can see these students have aligned themselves up by height. You can see that there are some very, very tall students on one end, and very, very short on the other end, and more people in the middle, kind of the typical bell-shaped curve. But you can see that there's many different heights because of polygenic and, in that case, multifactorial influences. Skin color. Skin color is made up of a combination of mostly 3 different genes. They call them the A, the B, and the C gene. And depending on whether you have dominant or recessive alleles for those genes will depend on your skin color. So if you have more recessive alleles, your skin color is lighter. If you have more dominant alleles, your skin color is darker. And you have all the other combinations of skin color in the middle. Again, skin color being another one of those that is multifactorial, because obviously it's affected by what you eat. It's affected by exposure to sunlight and things like that. Eye color. Eye color is also polygenic. It's based on 2 genes, the A gene and the B gene. And these 2 genes have to do with the thickness of the iris and also the number of pigment cells within the iris. The more pigment cells, the darker, the thicker the iris, the darker the eye. And again, here you see a situation where if you get the more recessive genes, you get the lighter colored eyes, the blue eyes. And the more dominant genes, you get the darker eyes, and then you have all the other shades in the middle, so that each of these alleles is what you got from your mother and your father. So if you got 2 of these from your mother, 1 of these from your mother, 1 of these from your father, 1 of these from your mother, 1 of these from your father, and they're all recessive, you will have blue eyes. Likewise, if you got these from your mother and your father, for these 2 alleles, these 2 genes, excuse me, you will have very dark eyes, and the others are all in between. That ends the first part of this lecture on genetics. Please be sure to listen to the second part, and certainly let me know if you have any questions.